Now, I'm very proud. I'm very proud to introduce my work about zinc oxide nanoparticles exposure induce melanoma-like skin lesions in skin barrier deficiency mice and its mechanism. Now, first is the background. As we all know, zinc oxide nanoparticles is very popular, used in many kinds of cosmetic products because it has good dispersion quality, transparency, etc. So it is widely used in many cosmetic products such as sunscreen, etc. It is used to protect UV radiation. And it is reported that zinc oxide nanoparticles typically do not infiltrate the epidermis due to the strong barrier function of the hair skin. The hair skin can prevent the infiltration of the external materials. But in our daily life, there are certain pathological skin conditions accompanied by skin barrier injury, such as eczema or UV damage, etc. And in this condition, it will increase the risk of penetration by when we use metal oxides nanoparticles. And in our daily life, there are many kinds of skin diseases, such as atopic dermatitis, acne, etc. When we use at this condition, now great attention has been paid to examine the hazardous property of the zinc oxide nanoparticles when it is applied to the skin. So we want to know how about the skin with these diseases when it's exposed to zinc oxide nanoparticles. In our group, we have constructed CDC42 deficiency mice by keratin 5 promoter, and we have proved that the CDC42 is very is very important for the skin barrier formation, and the CDC42 deficiency is closely associated with the skin barrier damage. As a result, use this mouse model, we try to examine the hazardous effects of the continuous zinc oxide nanoparticles exposure. Now, next, first we use nanoparticles, nano-sized particles, and we'll try to investigate the influence of nanoparticles on this kind of skin, on this kind of mouse model for CDC42 knockout mice. And we sacrifice the mice after exposure for 4 days, 2 weeks, and 7 weeks. Now, we can see after exposure for the nanoparticles, the CDC42 knockout mice, the water content of the skin is very lower than other groups, treated or untreated. And we can see the skin change to dark when exposed to 2 weeks and 7 weeks. That means after 2 weeks and 7 weeks continuous zinc oxide nanoparticles are increased, skin pigment was observed. And maybe, and next, we use transitional electronic microscope to observe it further. And we can see in the deeper epidermis layer, there are many particles in it. Next, use the light microscope to observe the skin. We can see the skin, the epidermis layer are changed. And we can see when continued to treatment for 7 weeks, in the dermis layer, we can see some pigment granulars in it. And we want to know what happened for the skin when exposed to the oxide nanoparticles. So we use the transcriptome sequencing to check the change. Now, after screening out the transcriptome change, we can see the three genes the 3 genes has changed. One is for FKBP51 and the TRM63 will increase in the knockout mice treated with nanoparticles. And the TSP1 is decreased in the knockout mice treated with nanoparticles. And for the FKBP51, some research reported it is associated with the beginning and the progress of the melanoma. And for these 3 changed gene, we use the qPCR we also use the qPCR to identify it. Next, we use the marker of the monocytes to do the exam. First, for the TYR, we know TYR is one of the biomarker for the monocytes. We can see after exposed the nanoparticles, we can see the knockout mice. The monocytes is increased in the not only in the epidermis layer but also in the dermis layer. And for the DCT is another biomarker for the monocytes. We can see after treatment for 2 weeks and 7 weeks, the FKBP51 and the DCT double positive cell are increased. And this is next we examine the proliferation cells. Now we can see for this 2 time point, we can see the proliferation cells. We use KL67 to check it. Now we can see the proliferation cells are increased for the knockout mice after exposed 2 weeks and 7 weeks. But for the apoptosis cells, we use internal experiment to check it. We can see the apoptosis is decreased in this in the knockout mice after treated 2 weeks and 7 weeks. So now we primarily get the conclusion that maybe the experiment epidermal viral dysfunction model mice treated continuous with oxide nanoparticles develop melanoma-like lesions and induce the dysregulation expression of the gene associated with melanoma. As we all know, some studies have showed that the nanoparticles deposited intact organ can generate excessive oxidative stress. So we use CET, GSH, TSOD, and MDA to check the ROS change. Now we can see zinc oxide nanoparticles treatment significantly increase oxidative injury in the skin of the barrel dysfunction mice. Now, some reported have showed that the mantle nanoparticles could make ROS produce, and the oxidative stress have been shown to be involved in all stages of carcinoma and malignant transformation of melanocytes. And some research have reported that high level of ROS could make melanocytes transformation by activity NF-κB and other transcription factors to initiate a downstream stress response, including the anti-apoptotic pathway. So next, to investigate the mechanism, we calculated the human epidermal melanocytes in vitro. Now, after the exam, we get the result. Treatment with 5 microgram per ml zinc oxide nanoparticles for 72 hours. The cells validation and the FKBP51 expression was increased. And we can see the cell proliferation was increased at this condition. And but the cell apoptosis decreased at the same condition. And the next we check the NF-κB and the BCL2. It is an anti-apoptotic pathway signature for signature. Now we can see 5 microgram per ml zinc nanoparticles treatment for 72 hours lead to NF-κB activation, accompanied by BCL2 expression. So next we check the ROS. We can see at this condition, the ROS content was increased, and use the NAC an inhibitor for the ROS. We can see the NAC could inhibit the ROS increase treated by zinc oxide nanoparticles. To identify is the ROS from the nanoparticles treatment, trigger anti-apoptosis effect mediated by NF-κB activation. Now we do the Western blots to examine it. Now after a serious experiment, we can see the zinc oxide nanoparticles induce intracellular ROS, which trigger an anti-apoptosis effect, which is mediated by NF-κB activation. Now that's all for our research. Now thank you for your attention.